Welcome to science class. Today we are going to learn about the process of meiosis, which is a unique form of cell division that produces reproductive cells in multicellular eukaryotes. You inherit 2 copies of every gene, with each parent contributing virtually the same genes and the same amount of DNA. The only exception is the passing on and inheritance of the Y chromosome. For simplicity's sake, we're going to ignore the Y chromosome in this video. The result of fertilization is that we inherit 2 copies of every gene. A cell with 2 sets of chromosomes is called a diploid cell. In order to produce diploid cells, reproductive cells must only contain 1 copy of each gene, because they combine their genetic material. Reproductive cells have half as much DNA, half the number of chromosomes, half the number of genes, and they are known as haploid cells. But, and this is crucial, reproductive cells do contain 1 copy of every gene. The full set of genes for a human is contained in each reproductive cell. In the normal cell cycle, a cell with 46 chromosomes duplicates all its chromatids, then splits them apart, creating 2 new cells, each with 46 chromosomes. The collective DNA in those 2 new daughter cells is virtually identical to the original cell. But in meiosis, a cell with 46 chromosomes duplicates all its chromatids, but then divides them twice to ultimately end up with 4 cells that each have 23 chromosomes. All 4 of the resulting cells are genetically different from each other. We're going to go through that process step by step. Let's get started. We are going to use this cell to visualize all of the steps of meiosis. It only has 4 chromosomes, so it will be easy to track them and see how, in the end, this one cell produces these 4 cells. Let's talk about these chromosomes. There are 4 of them, but we want to think of them as 2 pairs of chromosomes. Each parent of this imaginary organism passed on 2 chromosomes to produce the 4 that we have here. The chromosomes are complementary. They each contain the exact same genes, albeit with many of them being unique alleles for those genes. For each gene that is on chromosome number 1, the same genes for the same traits in the same locations are found on chromosome number 2. Same story goes for chromosomes 3 and 4, albeit with separate genes from 1 and 2. Chromosomes 1 and 2 are said to be homologous chromosomes. They are a pair. Chromosomes 3 and 4 are also homologous. At first, the cell goes through interphase just like any other cell in the body. All of the chromosomes become duplicated, with 2 sister chromatids attached to each other near their center. Meiosis occurs in 2 stages, called meiosis 1 and meiosis 2, and the steps have the same names as in mitosis, just with a 1 or a 2 to indicate what point we are at. In any case, the events in each phase are very similar to mitosis. I will only cover what is different. During prophase 1, crossing over occurs. Remember how I said the homologous chromosomes contain the same genes in the same locations? What these homologous chromosomes actually do is they exchange part of their chromatids with one another. Genes are genes. The variations between alleles in genes are so incredibly slight that there's no real problem in exchanging them. This is part of what produces such variety among sexually reproducing organisms. Before crossing over, chromosome 1 only represented one parent's genes. Now it represents, to a certain extent, 2 individuals' genes. The full impact of this won't become obvious until the end, but keep it in mind. Next we have metaphase 1. The homologous pairs of chromosomes line up side by side during metaphase 1. During anaphase 1, where the DNA is pulled by the spindle to each side of the cell, the chromosomes remain wholly intact, sister chromatids still attached. This is called a reduction phase. Remember, an individual chromatid is considered 1 chromosome, but so is 2 chromatids when they are combined together. In mitosis, these 4 chromosomes would be split into 8 chromatids, which would eventually give each cell 4 chromosomes. However, Because of how anaphase 1 splits the DNA, we now have only 2 chromosomes on each side of the cell. So our 2 daughter cells produced at the end of meiosis 1 each only have 2 chromosomes. They are haploid. Telophase 1 in cytokinesis occurs, and there's really nothing to talk about there. But let's compare the result of meiosis 1 with mitosis. If these cells had divided through mitosis, there would be 2 cells with 4 chromosomes each. But because the chromatids did not separate, instead we have 2 cells with 2 chromosomes each. Now both of these cells will divide again, but they will not go through interphase. This means that the DNA is not going to be duplicated. We are going to split what we have right now in half again, producing 4 haploid cells, each with exactly 1 copy of every gene, rather than 2 copies. The stages of meiosis 2 and mitosis are basically identical. In anaphase 2, the chromatids separate. So where there once was 2 chromosomes made of combined sister chromatids, each daughter cell will now contain 2 chromosomes but made of individual chromatids. There we go. 4 haploid cells, each containing 1 copy of every gene. We can also see that these cells are genetically different. If crossing over had not occurred, then 2 of these cells would essentially be genetically the same, and the other 2 would be different from the first 2, but identical to each other. But because of the mixing of DNA between each parent's chromosomes, now there's a certain amount of swapped DNA in each of the 4 cells. This is partly what produces such genetic diversity among sexually reproducing organisms. Remember that we only pass on 1 copy of every gene, but we inherit 2 copies of every gene. However, during the S phase of interphase, all of our DNA is copied. This means that just before mitosis or meiosis begins, there are actually 4 copies of every gene. This is how meiosis can produce 4 cells with 1 copy of every gene in them. Now, meiosis in men and women is different. A lot different, actually. In women, meiosis 1 occurs during the fetal stage, before you're born. Then it shuts down, or rather pauses, until puberty. In any case though, females are actually born with all of the egg cells they'll ever have. The second round of divisions, meiosis 2, those only start occurring after puberty, and then they end at menopause. Technically speaking, an egg cell does not complete meiosis 2 until after it has been fertilized. But it gets more complicated. The cell divisions in female meiosis are not equal. Meiosis 1 results in 1 very large cell and another much smaller, non-functional cell called a polar body. The egg prioritizes the cytoplasm and majority of organelles to 1/2 of the split. Then after fertilization, the same sort of thing occurs, and the egg again splits into the functional egg, actually it's a zygote at this point, and another polar body. The reason for this is that the cell divides multiple times after fertilization, but it doesn't grow larger. It's like cutting a pizza. 8 slices of pizza is the same size as 1 pizza. The egg must have the bulk, resources, and energy within it to accomplish the initial mitotic divisions of embryonic growth, because implantation and the sharing of resources with the mother happens several days after fertilization. Then meiosis ends at menopause, along with the reproductive capabilities of females. In all, a human female will be born with around 400,000 eggs, but will only release 1 a month for perhaps 30 years, about 360 eggs. But in males, things are much more simple. Men don't begin meiosis at all until puberty, where it is a continuous process rather than a sort of monthly process. It also continues throughout life. Men can father children until the day they die. Meiosis also always produces 4 functional sperm cells from the 1 original diploid cell, and a lot of them. Somewhere around 20 million per day. Why? Well, that's a very long and complicated answer that has to do with evolution, not genetics. That actually concludes this video on meiosis. In the next video, we're going to begin exploring the structure of DNA itself. Thanks for watching.